Hello everyone, I want to thank you all for coming today. My name is Sarah Cole. I'm going to be talking about the effects of flavonoids and humic acid on soybean growth, and I'll explain what both of those substances are. First, I have flavonoids. These are biostimulants, which means that they just help the plant grow overall. They mainly help with photosynthesis and basically the green part of the plant. So it protects against UV radiation, and it actually attracts pollinators by making them brighter in color. It also fights against microbial compounds, which will help them, like their immune system, basically. And they also have light screening and pigment functions. Like I was saying earlier, it helps with photosynthesis, and it helps absorb the light in a way that should help them not be harmed by the light that it's getting. And it also helps create rhizobial connections in the soil, and these connections will help the plant get nutrients, more nutrients from the soil than they would without these connections. Humic acid is another important compound. It enhances decomposition in the soil, so that helps get extra nutrients from the organic matter in the soil. And it also outputs useful nutrients that the plant can use. It produces usable forms for them. And it also improves enzyme activities and protein synthesis, which is important in getting all of those nutrients and using them throughout the plant body. It also increases holding capacity and pH, and it regulates the pH in the soil. So this helps so that it doesn't get burnt out by acid, or so it's not too alkaline. And it also has metal binding abilities, which means that the plant won't have to uptake these metals, and these metals, including mercury and stuff like that, actually harm the structure of the plant. So these metal binding capabilities in the humic acid helps prevent this damage that these metals can do in the plant structure. So I chose soybeans to use for this experiment because they have such a high impact and they're such a big cash crop in the economy. So they produce about $115.8 billion a year in the global economy. So obviously this is a big thing to be testing on to make sure that we can grow them more efficiently in the future. They are a bean pod plant with about 5 or 6, maybe 6 beans in the pod. 5 is more typical, 6 is if they're really healthy. They're a legume, which means that they are a nitrogen fixer, so they put nitrogen back in the soil after they die. And like I was saying before, they're a popular cash crop. Along with corn, they're probably the top 2 cash crops in the world grown. And they're high in fiber and protein, so this is good for people with low meat diets, or people who are vegetarians or vegans that don't have the protein from the meat that they would be eating. So my experiment was a little tricky to begin with. I had 2 attempts. My first attempt failed, and it was just rocky to start off with. I couldn't get the soybeans in time, and my original plan was to plant them all in a 24-acre field, so the entire field would be covered. And in reality, that just didn't happen because there was a soybean shortage, and we just couldn't get enough soybeans for that. So I just decided to do 2 17-foot strips. One was a control and one was the experimental, and you can kind of see that up there. I labeled them too. The experimental group was sprayed with flavonoids and humic acid about 3 weeks after their planting. I had to make sure that they had their leaves up and stable enough to support the flavonoids specifically. And the humic acid was perfectly fine either way to be put in the soil or the leaves, but the plants were able to take them either way. As you can see on the left here, they were all eaten by deer. You can see them chopped pretty much 3 inches above the ground. That was how my entire test group and control group that's how they were demised. So that made me have to restart. So I restarted here at Cazenovia in Eckel 305, which is actually in this back corner here. I planted 64 pots of soybean plants to make sure that I had a decent enough group to test. So I had 2 trays for my control group and 2 trays for my experimental group. And again, the experimental group had the flavonoids and humic acid. And actually, it was the left side that had the experimental group. So here, these were the ones that were sprayed, and the other group was just left normal. I made sure to have 1 experimental and control on each tray to make sure that if there were any variabilities, that it kind of evened out. So like I was saying, I planted them on October 4th after my first attempt was destroyed. I applied fertilizer about 2.5 to 3 weeks later when I was able to get the stuff. So that was like October 20th and October 27th. First, it was October 20th. That was when I applied the humic acid, and then on the 27th, I applied the flavonoids, simply because I just couldn't get the flavonoids in time to do them all at once. So I was forced to do that separately. They had constant light from the growth chamber that they were in, and obviously continuous watering to ensure that everything was moist and ready in there for them. I measured the total dry biomass of these guys, and my hypothesis was that the addition of the flavonoids and humic acid would increase the soybean growth compared to those without these substances. My process included November 8th, a couple weeks ago. I took 20 plant samples from each group and I separated them and cut them just above the soil. So I took, like I said, 20 plants from the experimental group and 20 plants from the control group, and that included both of the trays. So you can see the before and after shot of when they were cut. My results were not what I was expecting. So this first one here is the dry biomass chart. So this is what I did here in Eckel. You can see here that the experimental group was actually a little bit more variable. The experimental group is over here. So on the right there, you can see that that was a little bit more variable than the control, which I wasn't expecting because I was really expecting the experimental group to just be producing more. I thought the biomass would be bigger, but as you can see there was a huge variety. The lower outlier was really a lot lower than I was expecting it to be, and it was much lower than the control group was. And it was interesting for me to see that on my original attempt, that it was kind of the same thing. I was able to collect a couple leaf samples from each group before their demise. So again, the treatment was on the right and the controls on the left. So you can see that the results were just a lot more variable than I would have expected. The control group is pretty compact in how tall they got, and the treatment group was either really, really small or really, really large, which I just wasn't expecting. But their means were the same, which was also interesting to see. So as I was saying, there was a lot of variability in my experimental group, so I had a lot of possible reasons why that could happen. First, I thought maybe the potting soil there wasn't soil microbes in the potting soil, and this is what the flavonoids and humic acid would really react with, so these microbial connections just could not be made with those substances. I thought maybe that would be a reason why that it just didn't work. I also was looking at the stem structure, and I found it very odd that these stems were very flimsy, and it didn't support the plant very well. They were very skinny, and like I said, the plant just wobbled to the side when it got too tall. And if you remember in my first picture of my initial attempt, the stems were pretty straight and they were very sturdy and they held the plant up. So I thought that was odd, and I looked at other aspects that might be affecting that as well. I thought maybe with my lab-grown plants that were happening here, the unnatural photo period would affect that. So I had these lights on in the growth chamber 24/7 for the entire month and a half that they were planted. And I figured maybe that had something to do with it. And I figured maybe if I did this again next time, I would do an 18 to 6 hour ratio. So it would be 18 hours of light and 6 hours of darkness to create a more natural situation for them. I didn't know if that would affect it, but in the future I would probably do that. I also didn't know if the substances were interfering with one another. Sometimes that just happens and there's really no explanation why. So I thought maybe in the future I would do some with just flavonoids, some with just humic acid, and then another group with both. I also thought maybe with the lab-grown plants, they were all doing well and they were all similar because they all had the same amount of nutrients, and use specifically nitrogen because that helps plants the most in terrestrial plants. So I thought maybe with the potting soil having that high amount of nitrogen, that just helped them all to be about the same size. And that's all.